start, you'd had some lunch and you were digesting your lunch, and we looked at some of the mechanisms that are used to digest carbohydrates and how they get to those monomers, the monosaccharides that can then be transported into the cell. What we're going to do is to look at what the next stage of the utilisation of carbohydrates, and in particular glucose, is in terms of the catabolic pathways in metabolism. So as you can see, there's quite a lot of learning objectives that we're hoping to cover in this session. So I'm not going to read them out, but hopefully as we go through you'll be able to see where these come up. And in fact I think probably what's going to be a bit more helpful, rather than going through the learning objectives at this point, is just to look at an overview of the lecture. So what I'm hoping to do is to give you an overview of the process of glycolysis. So that's stage 2 of the metabolism of sugars. So we looked at stage 1 in the first lecture. Let's move on to stage 2. And we're going to split that into 2 parts, into 2 phases. We're going to look at phase 1, which is where some energy is put into the pathway. And that energy is put in to basically to activate glucose, to enable the second part of the pathway to proceed, where we're actually going to see some energy return. So we're going to go from putting some energy in the form of ATP, which is a chemical really rather than energy, but we'll call it energy for now as it contains energy in the chemical bonds. But then we'll then see some energy output by more ATP being produced at the end of the process of glycolysis. It might seem that after about 10 slides the lecture's finished, because I'll have got to this point and it will seem like we've actually done glycolysis, which would actually be quite nice if we had, but there are some other things that I just want to talk about around glycolysis once we've got the overall picture. So we will look a bit at the metabolism of other sugars, so we'll see what happens to galactose and fructose and how that gets into they get into the process of glycolysis. We'll look at a bit of the regulation of glycolysis, because it's with metabolic pathways, it's important to know where like the pinch points in the pathway are, where the pathway can be regulated to control the flow or the flux through a pathway. So we'll look at that. We'll go on and we'll look at anaerobic glycolysis and under what situations anaerobic glycolysis is important, and we'll relate that to an enzyme, lactate dehydrogenase. Then the end's almost in sight at that point, and what we'll just do is to have a quick look at some important intermediates in glycolysis, and one of the things that we're going to look at as well is something called the pentose phosphate pathway that sort of branches off from glycolysis and then rejoins it, and it's important in understanding one of the I suppose reasonably common genetic diseases. So that's where we're actually going to be going over the next 50 minutes or so. So we're looking at this stage now in the catabolism of glucose. We've looked at the carbohydrates from the diet. We're now down to the point where we've got monosaccharides, and we're going to look how those monosaccharides are converted to pyruvate and in some cases to lactate. So this is called glycolysis, and it occurs in all tissue types. It's intracellular, so what we were looking at in the first lecture was extracellular, occurring in the gut. What we're looking at now is intracellular, and it happens in the cytosol, so the cytosolic process. I've tried to put in some links that you should be able to follow from your iPads to animations that help to illustrate what's going on in this process, because I actually find these processes are really useful to look at animations. So you should be able to follow those links and use them in things like your revision to get an overview of what's going on. So this is what we're we're going to be looking at then? Glycolysis. So this is the central pathway in carbohydrate catabolism. So it occurs in all tissues in the cytosol. It's got three important functions. So it's going to oxidize glucose, and in turn it's going to produce NADH, which gives us reducing power in the cell. There's also going to be some synthesis of ATP, so that's going to occur from converting ADPs to the diphosphate nucleotide into the triphosphate. And we're going to break down glucose from a 6-carbon molecule into 3-carbon intermediates. And we'll see that some of those are important in terms of other cellular processes. Things to look out for is that this is an exergonic process, as catabolism is. It's releasing energy. It's oxidative in terms of the glucose is being oxidized to be able to allow us to produce the reducing power of NADH. The carbon is preserved. So what I mean by that is we're going to start off with a carbon-6 molecule, a molecule that contains 6 carbon atoms, and we're going to end up with 2 molecules that contain 3 carbon atoms, and that's still going to be retained within the cell. We're not going to lose any carbon dioxide. We'll look how glycolysis can operate anaerobically and why that might be important for some tissues to be able to operate under anaerobic conditions. The main role, though, that we'll look at is that this is going to feed into stage 3, or the Krebs or the TCA cycle or the citric acid cycle, three names for the same thing, that we're going to look at next week. The bottom line of glycolysis is that you're starting off with glucose and with some inorganic phosphate and some ADP and some NAD. We're going to produce 2 molecules of pyruvate, so that's our 2 molecules that contain 3 carbons. We're going to have 2 molecules of ATP at the end, the net outcome of 2 molecules of ATP. So that's our energy carrier, and 2 of NADH, plus some protons and some water. So glycolysis involves 10 enzymes. You don't need to learn them. You don't need to be able to recite off every single enzyme, although you'll notice that one of the activities on Moodle for you to do does actually get you to name them. But if you don't get that 100% right, I'm not worried about it. But just for your information, we've got the 10 enzymes here. If you like picturing things in terms of chemical structures and what's going on, which I'm guessing that some of you might, having done A-level chemistry, and that's sort of like my background as well, I like knowing what's happening chemically. There's a slide at the end that shows you what the intermediates are. You don't need to know the chemical structures, but if it helps you to visualize what's going on in the process, then you might find that useful. Now, I said that we're going to divide it into 2 sections. So we've got phase 1, which is where energy is put into the system. That involves the first 3 reactions in the pathway. And then phase 2, which is where we get the breaking apart of glucose into 2 3-carbon molecules, and the release of the reducing power and the the production of the ATP is happening in this phase 2 region. So you might reasonably ask, why have cells got such a complex process? Why are there so many steps? Surely there must be a chemically easier way or more efficient way of actually going from glucose to smaller molecules and produce energy on the way. Well, the chemistry occurs more easily in smaller stages, and obviously that can happen biochemically in the cell because of enzymes that can facilitate the process and can speed up reactions that would normally take a long time without the presence of an enzyme to catalyse the reaction. By having so many steps, there's actually fairly efficient energy conservation, so you don't get loss of huge amounts of energy by sort of fewer reaction steps. And there's also a lot of versatility. So no metabolic pathway acts in its own, so there's interconnections with other pathways that we're going to see. There's the production of useful intermediates, and also some of the pathway can be used in reverse as well. And then perhaps one of them, well, they're all important, but equally as important is that the pathway can be controlled. So there's regulation of the pathway that can adapt to the cell's ongoing energy needs. So the individual enzymes can be upregulated, downregulated in their activity to either produce more product or to slow down the production of product. So let's look at the phase 1 of glycolysis first then. So those first 3 reactions, what are the key things that we need to pull out from this? Well, actually the first reaction is really important. So that involves the phosphorylation of glucose to glucose 6-phosphate. So that's the addition of a phosphate group onto glucose. This gives the glucose an overall negative charge. You've added a phosphate that contains a negative charge, that's onto the glucose. That prevents passage back across the plasma membrane. Now, glucose won't normally cross the plasma membrane, but if you remember, cells have got those GLUT transporters that will facilitate the flow of glucose across the plasma membrane. Now, if you were to be in a situation where you had high levels of glucose within the cell, the flow of glucose could change, and it could use the GLUT transporters to go back out of the cell. Whereas as soon as you've got a phosphate group on the glucose, that traps the glucose within the cell. It also plays an important part chemically because it increases the reactivity of glucose. So by adding that phosphate group on, that alters the overall reactivity of the glucose, allowing subsequent reactions to occur. So this first reaction then, glucose to glucose 6-phosphate, requires energy. So it requires use, when I say requires energy, it requires the use of ATP. So it's taking up some of the cell's valuable resources that it could use for other metabolic pathways, but it's making an investment. It's keeping the glucose within the cell. It's preparing the glucose for further steps in metabolism, and in particular in glycolysis. We also find that reactions 1 and 3 have a large negative Gibbs free energy change. So I think Claire went over that with you last week briefly about what makes reactions likely to proceed in forward reverse conditions. This large negative energy change means that the reverse reaction, going from the products of reactions 1 and 3 back to the initial starting materials, is energetically unfavourable. So effectively these reactions become irreversible. Reaction 3 is also what's known as the committing step. Glucose 6-phosphate can be used in other pathways. There are other ways for glucose 6-phosphate to come into glycolysis. It doesn't have to come through the phosphorylation of glucose. But there are also other cellular uses of glucose 6-phosphate. However, once step 3 has occurred, where you get the addition of a further phosphate group, So again, this is another ATP molecule being utilised to phosphorylate, make what's known as a bisphosphate. So now we've got a sugar that's got 2 phosphate groups on it. At this point, the glucose is pretty much committed to going down the path of glycolysis. So we've got an initial investment of energy that later on in glycolysis is going to be recouped. So in phase 2, so we've got this fructose bisphosphate that's then going to be, that in the early on in phase 2 is going to be broken into 2 3-carbon compounds. And these compounds are also interconvertible. So what we've got here, reaction 4 will produce these. They can isomerise between the 2 of them. But it's when we get to reaction 6 that we start to see some sort of payback now. So in reaction 6, we see some production of NADH. So we're starting to get some reducing power. So from that initial energy input, we're starting to recoup something. Then when we move on to reaction 7 and 10, this is where we actually get net ATP synthesis. So both reaction 7 and 10 are producing ATP. What we have to remember with both the production of NADH at reaction 6 and also ATP at 7 and 10 is that we now have 2 3-carbon molecules from our 1 glucose. So if you remember that there were 2 phosphorylation steps of glucose in phase 1, so 2 molecules of ATP we used. For each molecule of glucose, we've got 2 3-carbon intermediates passing through the pathway. And so as a result, for each glucose molecule, in fact, we're getting 2 molecules of NADH, 2 molecules of ATP, and 2 molecules of ATP here. So overall then, we're getting a net gain of ATP synthesis, because in phase 1 we're using up 2 ATP molecules. In phase 2 we're producing 4 ATP molecules. So we're 2 ATP molecules up per molecule of glucose that started. We also have another point here where we've got a reaction that has a large negative energy. So again, we have another reversible reaction. So that reaction to produce pyruvate is an irreversible reaction. So there are 3 irreversible steps in glycolysis, and these are points at which the process can be regulated. It's also worth remembering that should a cell be required to produce glucose, it can't just put glycolysis in reverse, because they're 3 barriers to glucose reforming from pyruvate along exactly the same metabolic pathway. So it's a bit like a one-way street. You can go down it, but if you want to go back to the starting point, you can't come back up the same way. There needs to be another route. Again, I've given you a link here. If you want to know in more detail what's going on, you can refer to textbooks. You can also look at that animation, and that shows you quite nicely the actual chemistry that's involved, but you don't need to know that level of detail. So this slide really just summarises what I've said. So we've got the investment in reactions 1 and 3 in phase 1 of ATP, but we recoup that in terms of the ATP that's generated in reactions 7 and 10, where each glucose molecule is producing two molecules of ATP in each of 7 and 10. So overall, we've got a net gain of 2 molecules of ATP. this process for producing ATP by adding phosphate onto ADP is known as substrate level phosphorylation. So the ADP is being phosphorylated to ATP. Okay, so that's a bit of biochemistry. Now let's look at something that's a bit more clinical. So, in cancer, as you know, rapidly dividing cells, the rate of glycolysis may be up to 200 times more than in normal cells. So, the technique of PET, positron emission tomography, can measure the uptake of glucose-like molecule that can be then traced within the body. And so this image just shows metastases of a cancer. So I think that's a colorectal tumour that's now showing up in the liver. And so that imaging is possible because of the abnormal uptake and usage of glucose within the tumour, which means that this tracer molecule can be detected. So this slide then is your overview of the basic mechanism of glycolysis. So I think I won't re-emphasize the points. These are all the things that I said as we started looking at glycolysis. These are the main features to be aware of. So what we're going to do now is we're going to just look around the process of glycolysis a bit more, and we'll see other bits of interest around glycolysis. So glucose is the main sugar that we've been looking at, but we've also said that we get fructose and galactose in our diet. So if these are going to be utilised, then there must be a way of integrating them into the carbohydrate catabolism that's going on. There must be a way of getting some energy from the galactose and fructose as well. And hopefully what this slide shows is that there are ways in which galactose can get into a form that can then enter the pathway of glycolysis and give us either pyruvate or lactate at the end. So again, the processes that we're going to look at are all in the cytoplasm. So let's consider fructose then. So if we have sucrose, if we have sugar in our coffee, if we eat a lot, or if we eat processed foods, there's bound to be sucrose added to it. So that's our disaccharide of fructose and made up of fructose and glucose. So the mechanism for metabolizing fructose is an enzyme called fructokinase that adds a phosphate group onto fructose. So very soon we see phosphorylation of glucose. Here we have phosphorylation of fructose. And then the enzyme aldolase that produces the 2-glyceraldehyde 3-phosphate that is one of those molecules, one of those intermediates in glycolysis. So 2 enzymes gets us to an intermediate in the glycolysis pathway that can then be used. So if we're looking for clinical importance in this, so there are 2 genetic diseases. So essential fructoseuria is when there's missing fructokinase. So if there's a a mutation in the fructokinase gene, no fructokinase is present, that means that this first step can't occur, so you don't get any fructose 1-phosphate. The result of that is that fructose it ends up in the urine and is excreted. So there's no clinical signs with that. In itself, I think I'm right in saying there's no particular danger with that. In the case of fructose intolerance, then the second enzyme here, the aldolase, is missing. This is more serious because the fructose 1-phosphate has been produced. You get then a build-up of the fructose 1-phosphate, and this can cause liver damage, and so the main treatment for that is to try and remove fructose from the diet so that the body's not overloaded with fructose. So there are mechanisms for getting fructose into a form that can be metabolized. There are relatively rare genetic diseases that occur when there are issues with those enzymes. How about lactose? Certainly in the West we tend to have a lot of milk and other dairy products in our diet. So lactose then, this disaccharide made up of galactose and glucose. So galactose is mainly metabolized in the liver, and again we have a kinase as the first step. So sort of a pattern emerging here, phosphorylation of the sugar, keep it inside the cell. And then there's what I think is rather a neat way of utilizing the galactose. So galactose comes in contact with this galactose 1-phosphate uridyl transferase. That's a bit of a mouthful. Basically what's happening is this enzyme allows this nucleotide, so if you think you've done RNA, haven't you already, and you know that about the U nucleotide in RNA. So what we've got here is a U nucleotide with glucose that in the presence of this enzyme, it will switch the galactose for the glucose. So galactose comes out from the reaction, but glucose 1-phosphate carries on. Now, glucose 1-phosphate is the product of our first reaction in glycolysis. So that glucose 1-phosphate can enter glycolysis and be used in glycolysis. But there's then a second enzyme that can take the galactose, and that basically causes a transformation of the chemical bonds in the galactose to glucose, so this recycles. So with the help of the two enzymes, you've got this sort of circular pathway that allows galactose to come in, glucose to come out, because the galactose is converted in this process. This has also clinical importance. So the genetic disease galactosemia, which occurs approximately 1 in 30,000 births. So here, where you've got a milk-rich diet in infancy, there's an inability to utilise lactose. All three enzymes involved can be affected. The galactokinase deficiency is relatively rare, as is, I think, the epimerase defect. In the galactose kinase deficiency, then you get accumulation of galactose. So the problem with that is that if galactose is present, it can enter other pathways. The key part is that the galactose can get converted to galactitol there, and then that's no longer able to be metabolized by the pathway that other sugars pass on through if they go through this particular intermediate. The more serious galactosemia occurs with mutations of the uridyl transferase. Because here again we've got the situation that the galactose can be phosphorylated, and so it's within the cell, particularly the liver, but then there's no pathway to get rid of it. So without that ability to switch it for the glucose, which would normally feed into glycolysis, you get accumulation of the galactose phosphate. So again, the best treatment for this is just to remove lactose from the diet so that you don't have the galactose to deal with. This is, you might want to check this in MGD, but I think it's inherited in an autosomal recessive manner. Okay, so that was a bit of a diversion then about other sugars and what might happen in the case of enzyme deficiencies involving those other sugars. Let's go back to the main part of the pathway and think about how glycolysis is regulated. So we've already identified that there are 3 reactions that are essentially irreversible, and they tend to be the points in metabolic pathways where activation occurs. So they're the most useful sites of control. In MGD, you're going to look at enzymes and you're going to talk about a type of enzyme called allosteric enzymes. Now, this is jumping ahead a bit. Allosteric enzymes are ones that have 2 sites. Most enzymes have what's known as the catalytic site, which does the business of the enzyme. So it takes a substrate will fit into it neatly, the protein will facilitate the rearrangement of chemical bonds so that the product can leave. Allosteric enzymes also have further regulatory sites. So it's where other molecules can bind to the enzyme, and allosteric is, forgive me if you're Greek and I'm actually getting this wrong, but as I understand it, allosteric in Greek means allo is other and steric is site. So in these enzymes, a regulatory molecule can bind to the regulatory site, and that causes a change in the structure of the enzyme that affects how well the substrate is converted to the product. So these allosteric regulators can either be activators or inhibitors. So you can either get molecules that can activate the enzyme to make this reaction more likely to happen, or they can inhibit it, which makes this reaction less likely to happen. So as it's shown in this diagram then, if you get activation, then you get an enzyme that can't really fit with its substrate well, so it's not very efficient. But as soon as you get that activation, the shape changes and the reaction can go ahead well. Whereas in this case with an inhibitor, the presence of the inhibitor changes the shape so that the substrate can't fit so well into the catalytic site and produce the product. So what we're going to see is that some of the enzymes in glycolysis are regulated by this sort of interaction, by their allosteric enzymes. Also, chemical changes can change the efficiency of enzymes. So covalent modifications, the addition of chemical groups to the enzyme, can affect how well it works. And a commonly used form of regulation is phosphorylation or dephosphorylation. So by adding a phosphate group can sometimes significantly increase the activity of an enzyme, whereas with other enzymes, the addition of a phosphate group can decrease the activity. Okay, so that's all a rather long-winded way of getting round to the fact of we've got our three sort of like pinch points in glycolysis pathway, our three irreversible reactions, and they can be regulated. So the first reaction, the hexokinase, so the production of the glucose 6-phosphate, that can be allosterically inhibited by the glucose 6-phosphate itself. So if you get build-up of glucose 6-phosphate, that will attach somewhere else on the hexokinase enzyme, and it will reduce the activity of the hexokinase reaction. The phosphofructokinase, so the enzyme that is catalyzing this part, where we go from what's fructose 6-phosphate to the fructose 1,6-bisphosphates, where we have the addition of the second phosphate group on this one. This is a good example of what's known as negative feedback or feedback inhibition. So this enzyme, the PFK, which if you were doing a biochemistry degree, you'd probably spend a whole lecture studying because it's such a good example of this type of regulation. It's sensitive to our ATP. So if ATP is produced further down the pathway, as will happen at step 7 and step 10, and the cellular levels of ATP increase, the phosphofructokinase, PFK, will be inhibited, and it will slow down the flux, the flow through that pathway. So it will slow down the production of ATP further down because it puts the brakes on at this point. On the other hand, if levels of ATP have dropped within the cell, what occurs is there's an increase in the amount of AMP, so the adenosine monophosphate. So that acts as a positive regulator, so that's a sign for low cellular energy, and if there's a large amount of AMP present within the cell, then that activates the enzyme and so produces, increases the activity of the phosphofructokinase. So this is quite important, for example, in muscle cells, where if the ATP is getting depleted because the muscle's doing a lot of work, it's burning up the ATP, and as a result, ATP levels drop, AMP levels increase. That gives the cell a signal to increase the flux of glucose passing through glycolysis to actually produce more of the product here so that the pathway can produce more ATP. That's not the only regulation though. I mentioned that phosphorylation or dephosphorylation, adding or taking away phosphate groups, can also regulate these enzymes. But in this case, the PFK can also be activated by dephosphorylation. So in the case of high insulin relative to glucagon, which you're going to study later on in this unit, that actually results in dephosphorylation, so an increase in the activity. And similarly, you've got the pyruvate kinase is also activated by dephosphorylation, and again related to the relative release of insulin or glucagon. I'm going to go on and mention one more point of regulation. And what I'm going to mention is what's going on at reaction 6. So if you remember, reaction 6 is where we get the production of NADH. Because if levels of NADH get high, that also results in an increase in glucose 6-phosphate, because high levels of NADH slow down this step, and there's a build-up which results in high levels of glucose 6-phosphate. So this is a case of what's known as product inhibition. So the product, NADH, when it's in high levels, starts to slow down step 6. And so that inhibits glycolysis. So why might we be interested in that? Well, you start to ask the question, what if the NADH isn't actually consumed? What if you get a build-up of NADH, and what then happens to glycolysis? Okay, so sorry, there's a lot of text on this slide, but let's work through it. So 2 molecules of NADH are produced per molecule of glucose, because although it's one reaction, we've got 2 3-carbon intermediates going through. That means that if we're going to produce 2 molecules of NADH, we need 2 molecules of NAD to act as the substrate. We need two molecules of oxidized NAD to be able to be reduced to the NADH. However, in the cell, the total amount of NAD and NADH is constant. It's not that NAD gets produced by another mechanism. You've got a balance of NAD and NADH, and there's a total amount. So if you have very high levels of NADH, then you're going to have low levels of NAD. Normally, in most tissues, NAD gets regenerated in the 4th step of catabolism. When we go on, we're going to look at oxidative phosphorylation next week. Normally, the NADH reforms NAD. However, there are some exceptions to that. So, for example, red blood cells don't have mitochondria. So mitochondria is where the 3rd and 4th stages of metabolism occur. So actually glycolysis is all that red blood cells have. Also, the 4th stage of metabolism requires a ready supply of oxygen. However, if you're doing strenuous exercise, the oxygen can easily be depleted, and so the muscle needs to act anaerobically, which again means that you're not getting that conversion back in the later stages of metabolism of NADH to NAD. So you're faced with a build-up of NADH and not enough NAD to allow the glycolysis to continue. So there has to be another way to try and regenerate the NAD, but that will allow for anaerobic glycolysis that's required in red blood cells and, say, vigorously in muscle that's under strenuous exercise. And that pathway is provided by the enzyme lactate dehydrogenase. So lactate dehydrogenase can convert NADH back to NAD. So it can oxidize the NAD from the reduced form back to the the oxidized NAD. So that will replenish the levels of NAD. That will in turn allow further glycolysis and further ATP production, whether that's in your red blood cells or whether that's in some muscle that's under stress. So the chemical reaction that goes along with that is the conversion of pyruvate to lactate. So you're now producing, rather than pyruvate being the end point of glycolysis, that's going to then feed on into stage 3 and stage 4 of metabolism, you've diverted from this. You've now got lactate. That in turn potentially causes a problem because you've then got a build-up of lactate. So the lactate has to go somewhere. So lactate that's produced by, say, our red blood cell that hasn't got the ability to use oxygen phosphorylation, or our exercising muscle, gets released into the blood. Once it's in the blood, it can get to the heart and the liver, where exactly the same enzyme, the lactate dehydrogenase, can actually convert lactate back to pyruvate, and with it convert NAD to NADH. Now the liver and the heart are always well supplied with oxygen, so aerobic metabolism isn't a problem for them. So that means that the NADH can readily be used in the cardiac muscle and within the liver. So the circulating lactate can be removed by those organs. They can then be used further for the catabolism within those organs. So in the heart muscle, end up as carbon dioxide. The liver can actually use the pyruvate for producing glucose. So just to illustrate about lactate production, so under normal circumstances, the production of lactate is relatively low, 40 to 50 grams in a 24-hour period. However, under strenuous exercise, that increases to almost the same amount, but within 5 minutes. So you've got quite a big difference between the amount of lactate that's being produced at rest compared to the amount that's being produced under strenuous exercise. There are also some pathological situations related to heart disease and the cardiovascular system that also result in lactate production. So, now then, we're looking at a slide that's talking about how the quantity of lactate within the plasma, so within the blood, is affected. So, the first two we've touched on already. So, depending on the metabolic status of the individual, there could be more or less production of lactate, and so under strenuous exercise, your production increases. There's also the counter mechanism in the heart and the liver to actually utilise that lactate. So that removes it, that acts as a balance. It's also possible to, the kidney will also remove lactate from the bloodstream. So usually the blood concentration of lactose is really, lactate, not lactose, is fairly low. However, if it increases, as it says on the slide there, to between 2 and 5 millimolar, then you're in a state of what's known as hyperlactemia. So you've got a raised above normal level, but what it means by saying below the renal threshold is that it's still at a level at which the kidney can facilitate its excretion. Where there's an issue is if the lactate rises above 5 millimolar. So that's above the quantity that the kidney can deal with, that the kidney can excrete. And so you're in a situation where you get a decrease of the blood pH. So that can go beyond the buffering capacity of the blood that normally holds your blood pH around about 7.4, but that buffering capacity can be exceeded if you have too high levels of lactate. Right, I'm afraid there's a lot of biochemistry here. I just wanted to point out that we said that one of the roles of glycolysis was also to produce small molecules that could be used in other metabolic pathways. So, in this case, one of the molecules produced in glycolysis can be used as a precursor for lipid synthesis. So the production of triglycerides and phospholipids that are important in the plasma membrane of the cell. So that's just one example of how glycolysis can cross over into other pathways. And then in the red blood cell, there's a regulation of the release of oxygen that's mediated by this chemical, the 2,3-bisphosphoglycerate. So that can again be synthesized from one of our intermediates from glycolysis. So again, we've got here a crossover with another pathway, so affecting the affinity of oxygen for hemoglobin in the blood. It actually favours the hemoglobin to take up a conformation that makes it more likely to release oxygen to tissues. And then our final diversion from glycolysis that we're just going to look at is something called the pentose phosphate pathway. So the pentose phosphate pathway occurs when we're in a glucose-rich environment. When there's a lot of glucose, glucose 6-phosphate builds up, and that can be diverted into this pathway. Now, often glucose 6-phosphate in liver and muscle will be used to produce glycogen. However, if there's more than is required for glycogen, this pathway can also be used. And what happens is the glucose 6-phosphate is siphoned off. It's used to produce NADPH by converting NADP to NADPH, so it's getting oxidized. And that NADPH can be used for anabolism in the cell. The byproducts of this are 5 carbon intermediates, and also 3 carbon intermediates that actually feed back into glycolysis. So apart from the loss of some carbon dioxide, most of the carbon actually works its way back into glycolysis. So again, this is occurring in the cytoplasm, just like glycolysis. The first stage then is what's known as an oxidative decarboxylation. There's an enzyme called glucose 6-phosphate dehydrogenase, and that is the first step that also that catalyses the production of 6-phosphogluconolactone from the glucose 6-phosphate, detail that you don't need to know, which can in turn be converted to a 5-carbon sugar with the loss of carbon dioxide. And the key thing here is the production of NADPH, so increasing the amount of reducing compounds within the cell. The second part of the pentose phosphate cycle is a rearrangement of our 5-carbon sugar. So this actually involves 3 5-carbon sugars that will produce, in the end, 2 fructose molecules and 1 glyceraldehyde. So, key things about the pentose phosphate pathway: there's no ATP production that occurs in this. There is loss of carbon dioxide, so that means it's irreversible. That carbon dioxide is then lost to the process, so you're stuck with the 5-carbon sugars. And overall, the pathway is regulated by that first enzyme in the pathway, the glucose 6-phosphate dehydrogenase. So the production of NADPH in the cell cytoplasm is important for its ability to take part in the synthesis of lipids and also in maintaining the structure of proteins. So this is key in red blood cells, where this is the main source of reducing power in the red blood cells, and without the reducing power, red blood cells become very susceptible to oxidative stress, so chemicals that can cause oxidation. Another important feature of the pentose phosphate pathway is the production of the 5-carbon sugars. So this is particularly important in rapidly dividing cells that need to maintain their levels of ribose, deoxyribose, for producing nucleic acids. So one of the more common enzyme deficiencies is a deficiency in the glucose 6-phosphate dehydrogenase enzyme. So there are multiple enzymes, multiple mutations that can, or polymorphisms I should say, that lead to this deficiency. It's found particularly in areas where malaria is or was endemic, because there's a process that goes on that actually means that people with malaria actually have a slight survival advantage if they have this particular enzyme deficiency. So the key role, though, in of the glucose 6-phosphate dehydrogenase, and so what doesn't occur in an individual who's G6PD-H deficient, is that there's an inability to keep proteins in a reduced state. This can lead to aggregation of proteins, which are seen as an aggregate called Heinz bodies in red blood cells, and also increasing susceptibility of the plasma membrane of the cell to lipid peroxidation. So that